to our symposium. I'm very pleased to introduce our first speaker, Guilherme Azevedo, who's going to be speaking about integration of micro and macro evolutionary models, sheds light on the speciation of cave spiders.
So thank you very much for being here after lunch. And this was part of my postdoc at SDSU, one of my projects where we tried to understand evolution of cave spiders. We developed this new approach to understand the evolution of cave spiders. We know that caves, they are features of karst environments, and those habitats are very important for maintaining healthy and diverse ecosystem. And so they provide water and provide resources for many, many animals, and humans are among the animals that use that. So you can see here the limits of Edwards Aquifer in the lines in black. They provides water for millions of people around San Antonio and Austin in Texas. And of course, we have like a lot of caves there, and many species that are unique to this area and adapted to caves. So it's important for us to understand the diversification process in caves, so we can, of course, have better knowledge about biodiversity, and also we can inform conservation. And one of the questions that we try to ask for, evolution of cave species is how the adaptations or the troglomorphisms impact the chance of a species to survive, to adapt, or to evolve, or to vanish over time, and how we can describe and understand descriptive diversity in caves. We know that state-dependent models, they provide a way to test that, so we can see how a trait, how the speciation rates associated with different states of the trait, how this trait influences the extinction rates, and also how the rates of transition between these two states on a tree. They're very useful, very nice, but they do not incorporate a process like the formation of populations, and that can be very important for caves because the species in caves, they are naturally fragmented into small populations. For that, we have the protected speciation model. So in this model, we have instead of species tree, we have population trees. So each branch represents a population on this tree. So the splitting represents a population formation, a population fragmentation rates, as you can see on your left side. And then this is the beginning of the process of speciation, but not the speciation completing itself. So for that, so after that, the species can go extinct or they can speciate. So the speciation process can complete on a branch. So the speciation model as an extended process on the branch length. So if you have, for example, the speciation completing on these two branches, the blue and the orange, we have 3 species of the tree, right? So we thought how we could understand the influence of traits on this speciation completion rate. So we developed an approach that we call the trait-dependent speciation model. So in our approach, we first used like an SSC model. It could be anyone. We use a HISI. I'm just representing here in the figure like a normal BC, but you can use anything. So you can estimate patterns. So you use that in your population tree, and then you can estimate the population fragmentation rate, the population extinction rate, and the transition rate between those traits. And so you can use this transition rate to sample histories of the trait history. That is, we can augment the data so you can have the tree with branch lengths and the state on the tree. On each branch, right? And also we can estimate the number. We can sample numbers of speciation events happen in this in each branch of the tree, and we use for that we use Poisson distribution. So we can have 1 or more 0 populations of speciation completion events happening, and this distribution is given by the branch length and the speciation completion rate, right? So we know that we must have we don't observe any speciation event in branches connecting, branches connected population of same species, right? So for example, everything that's blue there, we know that's 0, so we clamp that to 0. That's observed 0 speciation event. And calculate the probability given a branch length, the completion rates, and this is species assignment, so we need the species assignments to know which population are assigned to each species, right? And also, we can calculate the probability of one or more speciation events on branches connecting populations of different species. So we know that that must happen. So that those things constrain our search for this speciation history. For example, we know that this cannot happen because we have a speciation event on this, the ancestor of population 5 and 6. You know that population 5 and 6 is the same species as population 3 and 4. And I know it's a paraphyletic species, and it's fine. The model is fine with that, and I'm fine with that. And then we have our augmented data. So the speciation completion rate is determined by the state on each brand. So we have like two different speciation completion rates depending on the state of the brand. So with that, we can approximate the posterior distribution of the state-dependent speciation rates, the trait history, and the speciation history given the tree with branch lengths, the state transition rates, and the species assignments. We did that, we implemented that in RevBase, and we apply that to our system. This is a subgenus of the genus Sicurina and the subgenus Sicurella that represent one single invasion to caves, one transition to cave habitat, and they diversify into 76 species. And although they represent just one transition to caves, they adapt different to the cave. So we have different morphological adaptations to the cave that we are measuring as strugglomorphic index. This is shown to be a bimodal distribution, and we can put that in two states. So we're calling it the low trogloomorphic and the high trogloomorphic. And they have different natural history. So low trogloomorphic, they are usually like not so deep in the cave. They can explore more the entrance of the cave or a little bit in the middle. But the high trogloomorphic, they are very deeper in the darker parts and small parts of the tree, of the cave, sorry. So we ask, are highly adapted cave species evolutionary dead ends? So are they faded once they have these modifications? Are they faded to extinction? So if so, using our approach, we could expect that extreme specialization is not reversible, so they cannot go back to low trogloomorphic index once they are higher. We can also expect like a higher population extinction when they are very specialized because we have like smaller populations, they're so adapted and sensitive to habitat change. And also high population formation because since they are very specialized, they cannot move too much between the caves, so we structure very, very fast. But regarding the speciation completion rate that we are adding right now, we expect a high speciation completion rate on the low trogloomorphic, the purple one, because you know, it's been shown for spider they can, especially through sympatric speciation, through disruptive selection of habitat use in the cave. So some can be more close to the entrance, the other dip. So they still have room for evolving and changes. So we have like some selection is still high in the rates of speciation completion rate. So that's what we expect. And we run our analysis in our data, and we found exactly that extreme adaptations to caves are not reversible. As you can see, we use reversible jump to test the hypothesis of different models, and the reversible model is unlikely. And if you see the distribution of posterior probability of the posterior distribution of the rates, you can see the high TI to low TI are very, very low, and you don't see that on the tree, on the trait evolution. The same, regarding the extinction rates, we found that it's higher in highly trogloomorphic species, in high trogloomorphic populations. So the same, this model is better with two rates. You can see the red represent higher rates, and it's somehow in concordance with high trogloomorphic species, populations. And we also found that fragmentation is higher in highly trogloomorphic species. Sorry. So we found the extinction is high, or the population are not reversible, and then the extinction is high in highly trogloomorphic species, right? And also we found that the fragmentation is higher in highly trogloomorphic species, the same way. As you can see, the reversible jump showed that this model with different rates is better. But regarding the speciation completion rate, we found that the model with equal rates, it's actually better. So there's no influence of the traits on the speciation completion rate. So we can see the distribution of the traits, the rates, they're very similar. And if you look at the tree, you can see some variation, but the variation is not significant. So we know that the acquisition of highly trogomorphic traits are reversible and increase the population fragmentation and extinction rate as we expected, but not the levels of trogomorphism are not affected by the rates of speciation completion rates. But why? So it could be that the rates are similar or equal because the sympatric speciation, as we thought, are not common. They do not happen in this group. It could be that high trogomorphic species, can somehow evolve in sympatry through the selection of traits that we're not measuring with this index that we are using. It also could be that the population is so small that the fixation, the mutations that are incompatible fix really fast also. So a combination of these or maybe other things that we don't know, but we know that these have consequence, right? So answer our questions like maybe they're not completely dead ends, so they need time to speciate, and they need more chance to speciate, so it needs more population. So the consequence for the conservation implications for that is that we need to make an effort to have like more population, to preserve more population of this highly adapted species, so we can have the chance for them to speciate. This model can also be used for species delimitation, similar to the delineate. You can calculate if you have new populations that you think are new species. You can calculate the probability of speciation rate happening in the branch or not. In this case, no probability of speciation. So the take-home message here, I would like to use this code that we should start living with cars rather than living on or in cars. So with live is something you need to know better. This is valid for any environment, and these models that the unit of micro evolutionary models can help us to do this better, and we need more models. We need to test these models. We need simulations to understand how they work and to apply them to different systems so we can understand better this process in between micro and macro evolution. With that, I would like to thank you for being here, especially if you had a lot of poutine during their lunchtime and you're still here and awake. I really appreciate that. And thanks for the funding agents and our friends that help with this presentation. And I'd like to know that San Diego State University stands upon the lands of the Kumeyaay people, and that's where our work was done. And thank you very much. I'll be glad to take questions. Yes, I do, and some of the rates are explained by no variation. But that's the thing. The hidden states is only for the part of the HEC. We don't have hidden states on the spatial completion rate, and I'm not sure if the data would be have enough power to. Yeah, I try with like more states. Even if I have to use more states, usually the distribution, the posterior is given by the prior. So I don't know. It would be nice to see if I can implement like a hidden state on the speciation completion rate, also to account for another variation, because there is variation in general in the tree. Thanks.
We have time for another question. No?
Hey.
Very interesting, of course. I am having trouble with the speciation completion rate. Is it at later genetic rate? Where do you measure in the model?
It's at any genetic rate. So it's just the probability of completing depending on the branch of the rate. So it's modeled as like a state happening in the branch. So how many times the speciation complete in that branch? That's why we use Poisson distribution. So we just want to know how many speciation events happen, the chance of the speciation event to happen in that branch.
Okay, thank you.
Thank you.
Excellent. Thank you very much.
Thank you.
Oh, shoot. And our next speaker is Sean Anderson, who will be talking about tracking changes in the stability of post-zygotic isolation.
Yeah, thank you, everyone. Thanks for being here. I'm Sean. I'm a postdoc working with Dana Matute at UNC Chapel Hill. Thanks, Roger and Jonah, too, for putting this together. This has been really an interesting symposium. I like the concept or the question of speciation completion. And one thing that I think is interesting about it is our idea about what speciation completion means has changed a lot in the last 20 years in particular. So a question, when is speciation complete? And the idea for most of the 20th century after the modern synthesis was that it's complete when taxa establish sympatry, right? So the idea is the taxa speciate allopatrically, they come together in sympatry. If they remain and they don't collapse into a hybrid swarm, then speciation is complete because you have reproductive isolation. And this is, of course, a very logical view. I'm not sure it's quite the view we have today. I would say it's arguable. Genomic data obviously have changed a lot. And I think one of the best ways to illustrate that is to look at what the consensus was right before genomic data came online. So this is from Coyne and Orr's book in 2004, this exhaustive survey of the literature. And they were thinking about this idea of how we consider speciation being complete. And they said it mostly makes sense, but there's a couple cases where it doesn't quite work out. The worst case of all to them was oaks. Because oaks are sympatric, they look very different from one another, but even with the data available at the time, it was clear that some genes were getting across. All right, so this would sort of blow up that idea of what we think of as speciation being complete. But it was okay, they thought, because our guess is that morphologically distinct taxa showing rampant gene exchange at many loci will be rare. Right? And so that was the cutting edge view right before genomic data hit. And then, of course, genomic data hit, and we know that that's not the case. That's almost every week papers come out showing lots of gene exchange between morphologically distinct taxa. So, and it's not just Heliconias. So you've got in African cichlids that are sympatric and morphologically distinct, lots of gene flow. Between warblers in North America, easily identifiable, lots of gene flow going on. Plumage genes and little else separate their genomes. And it's not just animals, and plants you have it as well. In Mimulus, you have extensive gene flow between taxa, including taxa that are strikingly different in their vegetative morphology. So the concept of speciation being complete when sympatry is established, I'd say in the genomics era, is more questionable. And I think you can make an argument either way, but it is arguable. It's not quite clear anymore. Today, the way that I see it, and the way that I read the literature, is that people tend to think about the completion of speciation in terms of two axes. One is the extent of independent evolution across the genomes of the two lineages, and the other is how easily it can be collapsed, how easily it can be reversed. And they're not mutually exclusive. One probably affects the other. So I'm going to be talking mostly about the reversibility of reproductive isolation, or the collapse of species when you have them. An important note is that species do collapse. And we've got several good examples now where almost in real time you can see speciation collapsing. And this example of sympatric stickleback fishes, you see in the 70s these two distinct morphs corresponding to the benthic limnetic morph. And then later on in the 2000s, it's all just collapsed into a single admixed cluster. And this is thought to be the result of a change in the environment. There was no crayfish in the environment in the 70s, and there is a crayfish now, and it had a downstream effect that made it so that hybrid phenotypes weren't poor fitness anymore. So this would be an example of the collapse of an extrinsic barrier. But I want to think more today about the collapse of intrinsic barriers. So intrinsic barriers, intrinsic reproductive isolation is, it is just reproductive isolation that is not contingent on the environment. And it's thought to be stronger. And so again, Coyne and Orr, they say, as opposed to premating or extrinsic post-zygotic isolation, intrinsic isolation is likely to be permanent. And this concept, I think, is pretty popular. I've written it myself in the literature, but it's worth questioning to what extent this really holds true. So just to clarify, post-zygotic RI is just RI when hybrids suffer low fitness. Intrinsic post-zygotic RI is when they suffer low fitness, irrespective of the environment. And it can evolve in a lot of different ways. And there's a new paper out covering a lot of these that I recommend. The one I'm going to think mostly about, though, today is just classical DMIs. This is when you have independently evolving allopatric populations fixing alleles at different loci, and those alleles create incompatible combinations when combined. And so in the classic model that's in the textbooks, you have this biallelic genotype in allopatric populations. New mutations arise, they fix, And then when hybrids are formed, you're combining new combinations of alleles that haven't been tested. And so sometimes that can reduce hybrid fitness. So hybrid fitness is reduced, but is speciation complete? You have intrinsic isolation. Is speciation actually complete? Well, an interesting thing about this model is it's only complete if you allow no more reproduction or evolution to occur. So if you have, for instance, if you just take that F1 and you mate it with another F1, what kind of genotypes can you get in their offspring? So you can start recapitulating these high fitness genotypes. That's without recombination. If you include recombination, you can include almost all the genotypes that are on the high fitness ridge leading from the parent down to the offspring. And I'm not, of course, the first to point this out. A little while ago, Barton and Bankston noted this, and they said that genetic incompatibility is really the initial condition of the system. It's not the end. And you can see this in simulation. So this is from the SLIM GUI. I've got two populations here. The populations, or the individuals, are the little yellow dots. That they're yellow just indicates that they've got neutral fitness. If it goes red, it means they have poor fitness. I'm going to show you the last 10 generations of allopatry. So they're going through the last 10 generations of allopatry. Then sympatry kicks off, and you've got all these low fitness genotypes kicking in, right? But they don't persist very long. It does a pretty good job of getting rid of them. And so the question is, how do you stop that from happening? What needs to occur in order to stop this reversal of intrinsic isolation? And in order to get at that, I've been thinking about this concept in sort of two ways. The first is the effects that or what caused the alleles in the parental population to fix. Okay, so I just want to clarify that. In the classical DMI theory, it is irrelevant how the parental alleles fixed in the allopatry populations, because you don't need to know how that happened in order to explain why hybrid fitness is low. But when you're thinking about whether or not the alleles are going to stick around, you actually have to think about what happened in the parental populations. And so I've been working on this with Brian Lurch, who is a PhD student at UNC. And we've taken two tacks to this. One is looking at explicit models of individual incompatibilities, and another is looking at simulation models where they accumulate across the whole genome. And so what I'm showing you here is Brian's results from looking at the amount of time it takes to purge alleles based on how poorly fit they are. And what you'll see, I think, yes, is that the worse those incompatible alleles are, the faster they get purged out, right? Which is not altogether unexpected, but it happens very quickly. And something that we thought was a little surprising, it doesn't really matter if you have one or two. It's basically they're both gone very fast. And it doesn't really matter either whether the allele is expressed in the dominant or the recessive mode. They get purged out very quickly. So purging is extremely fast in incompatible genotypes, strongly reduced fitness. There's little difference in the purging time between 1 and 2 DMIs, and you have similar patterns when incompatibilities are recessive or dominant. So now this is also the case where the incompatible alleles are neutral in the native population. The thing I'm going to show here is what happens to population fitness at these different levels of selection. So the blue lines here represent highly unfit genotypes. And then the red lines represent genotypes that are less deleterious in the hybrid. So when they're highly unfit, they're purged immediately, and you immediately recover the population fitness. The key thing to note is just that when you have slightly deleterious incompatibilities, they stick around longer. You don't, because you purge them to the point that they're not being expressed all that often together, and so they don't leave. So what this leads us to believe is that when incompatibilities weakly reduce fitness, they're purged more slowly, and load persists. And so what this suggests is that the collapse of incipient reproductive isolation could actually be a source of load in populations that have a lot of geographic splitting. Okay, last thing here is just incompatible alleles when they're beneficial in the parental population. When they're beneficial, the only thing that matters is that the purging happens 10 times faster. Well, not 10 times faster, but almost. So essentially what's happening is the genotypes, are more fit if they express one of the beneficial alleles at a frequency of 1 than if they express both of the beneficial alleles at some incomplete frequency. So this is essentially haplotype competition that's going on. So one way that you can get rid of these incompatible combinations of alleles is not just recapitulating the parental, essentially reversing their evolution, but just having these haplotypes competed out when you have no pre-mating isolation. So it's another way that you can get rid of these things. So we have this problem. You can get rid of the alleles pretty easily. You can get rid of them fast when they're bad. One solution could just be to accumulate a ton of them, let a bunch of them build up. So it could be that when you have a short period of isolation, they come back together, you can easily purge the alleles. After a long period of isolation, they've got alleles, interacting alleles fixing across the entire genome. They're much harder to purge out. So this concept of at the beginning they're easily reversible, at the end they're highly irreversible. And if we can think about this in terms of what that change would look like, if you think of on the y-axis here, we have the reversibility of intrinsic isolation. And then on the x-axis, you have something like genomic divergence or time. At one point, it's going to be highly reversible. And at some point later, it's going to be less so. But we don't actually have any good sense yet just what that relationship looks like. If it's something like a straight line, if it's something more like a curve with some sort of inflection point. And there's a bunch of factors that could inflect where you might expect to see that inflection point. So we have lots of theory on the actual accumulation of DMIs, less so on how this change in their reversibility tracks through time. So it's an area to do some work just with basic DMI theory. Some of the relevant bodies of theory on this are, there's a lot. This concept has been thought about quite a bit over the eras. These bodies of theory differ very much, though, in terms of how much they consider the genotypes in the parents, which again is critical when you're trying to figure out if they're going to stick around. So to get to the part where we're looking at the accumulation of genotypes, I'm going to be using simulation models here. So we've got no divergent selection in this case. This is not a case where one allele is beneficial in one parent and not the other. So it's just the genetic effects that are driving down hybrid fitness. There's no pre-mating isolation. This is just supposed to be a good test of how good post-zygotic isolation is when it's intrinsic. We also have high migration rates. A lot of the testing of incompatible alleles is done with low migration. We want high migration rates here, including just random mating. The metric of interest is really the delay in species collapse. So if you have strong reproductive isolation and you throw things in together with no pre-mating isolation, it's an inherently unstable system, right? But if you can delay that collapse for a while, it might give these species time to find other means of persisting, involving pre-mating isolation and things like that. And we assume that the DMI effects are dominant. And so what I'll show you here, this is, the x-axis there is just the time in allopatry times 1,000. So at the 1, they've been in allopatric for 1,000 years. At the 10, they've been allopatric for 10,000 years. And then they come back together. And what you can see is that the collapse happens way faster over a certain period of time. So what's going on there? Well, it's not that actually the DMIs are being reversed. It's that they did their job. That accumulation made those genomes really impervious to gene flow. But as a consequence of that, when you have high mating, you have high haplotype competition. So one set of those genes is just beating out the other. It's essentially genetic competition that is preventing these things from co-occurring with one another. And you have, that process goes a lot slower when you have fewer of those alleles spread across the genome. And it also goes slower when you have higher recombination. Higher recombination rates allow more time to come up with these new beneficial combinations of alleles. You see, I'm just going to show quickly that you see a similar thing when the alleles were beneficial in the parents as to when they are neutral in the parents. And the things I want to highlight just at the end here, I'm running out of time, is just so far, intrinsic isolation collapses as a result of evolutionary reversal and as a result of haplotype competition. As DMIs accumulate, haplotype competition becomes the dominant mode that speciation collapse happens in. When DMI alleles are on their own or beneficial in the parental population, collapse occurs a lot faster because you just have greater haplotype competition. And the last thing I want to say is just that speciation collapse is also a continuum. Speciation is a continuum, but the collapse is as well. At some point, you get total genetic merging and essentially collapsing into a hybrid swarm. At other points, you get huge chunks of one of the species' genome vastly outcompeting the other, to the point where at some points, it's literally just one species beats the other out. So it itself is a continuum. And just last thing I want to mention is just that if anyone's interested in doing this stuff, I'm starting a lab this year in Atlanta, and I'd love to have people join and work on speciation with me. Thanks very much.
Hello, and thank you for being here. You're the survivors. You're still here after a fantastic 4 days. When is speciation complete and how does it progress? I think is one of these big questions in speciation research right now. And people have looked at it using empirical models, model organisms in natural populations, and theoretical work to address the question of the progression of speciation. colleagues and I are thinking about it in terms of dimensions and dynamics. And so the dimensions can be patterns of reproductive isolation, trait divergence, genome divergence, and so those are patterns, but they also, how do they proceed? Are there nonlinear dynamics for each of these dimensions, and how do they interact? But what we're really focusing on this talk is reproductive isolation and the nonlinear dynamics of reproductive isolation. And so foundational work generally showed us that reproductive isolation increased linearly or continuously with genetic distance. But more empirical work lately has suggested the role of tipping points, and that speciation might be more punctuated as you progress through stages of speciation. And so to look at reproductive isolation, I really focus on courtship behavior. And we did this in populations of red-eyed tree frogs that occur in Costa Rica and Panama. Their range is from Mexico to Colombia, but they show this really dramatic color pattern variation among populations. And we learned later that these have a strong role as sexual traits, which shifted our thinking to sexual selection through mate choice as a strong mediator of speciation in this group. And for that to be true, those conditions need to be met. There needs to be divergence in the sexual traits, variation in preference for those traits, really not a strong role of ecological selection, and that the females can perceive those male signals. And our work systematically worked through each of these, but what I'm going to talk to you about today is the preference data. And so we carried out a bunch of female mate choice trials. Some of them were choice trials, some were non-choice trials. We used robotic males, so we painted and sculpted red-eyed tree frog models to reflect the populations, had the advertisement calls being played with speakers, and did this for 8 field seasons. And we sort of framed our idea of how to measure reproductive isolation based on Bill Pyburn's 1967 work on the species. This beautiful painting shows the mating system of the frog, and I'm going to just walk you through it. First, there is the male will call. You see that inflated vocal sac, and then the female is approaching the male. They are in amplexus, so they have assumed the courtship position. But I want to point out this big black box, which is what happens here. What happens between that initial call and then getting together? That's a mystery that we're going to solve. And so then, but carrying on, the frogs now go down to the water, absorb water into the ventral sac. They climb up to a tree. They lay their eggs. The male fertilizes them externally. And so one of the questions is, what is happening in this 3-dimensional space? This is not a horizontal platform. They're doing this at night, which is hard to see, in a 3-dimensional way. And really all we knew about was that the males called to the females. And so I assumed the frog biology approach, which was phonotaxis, and that's really how people were studying mate choice in frogs, where you have a frog and a speaker, and then, you know, when they get close enough to the speaker, you celebrate it's a choice, and it's great, and you mark it down in your data. But that actually isn't what's working for our frogs. Instead, we realized that we needed to use female courtship displays as a metric of mate recognition and preference. And those displays we've now named a back display and a flank display. And I have videos to show you what these look like. Videos. How does it play? Okay, so here's a female approaching a male model. The male model's on the ground. And she's going to hold her body in a super awkward static position, showing her flanks to the model. And there's a really beautiful flank display. And then she tries again by showing her back to the male model. And if it was a male, the idea would be it would jump on. And so she's highly disappointed because that didn't happen. But it looks like phonotaxis because they was so close to that model. But I want you to know that that happens when they're upside down, 2 meters away, on a vertical substrate, on a horizontal substrate. It's just, it's not getting to the model that was important. We realized after years of watching them that they do those displays from quite a distance away. And so it was super obviously important to know what you're looking at. We discovered that females display to males, which, if you're a frog biologist, your jaw, I hope, is dropping, because that's unprecedented in frogs without parental investment. I can't think of another system where females try so hard to court the males, which then leaves this unanswered question: why do females even have to convince males to mate? It's an open question that I'm interested in. Because it's messy and it's complicated, it was really hard to try to quantify pre-mating isolation in the system because they didn't do phonotaxis. We had to really think about their biology and their natural history, but we did come up with an ethogram to be objective in our scoring. And so once we were sure that we knew what we were looking at as much as we could, we set out to ask about pre-mating isolation along as lineages proceed or progress. And so to do this, we looked at a bunch of pairs of populations that varied in color pattern and male advertisement call, and we measured female mate choice, and then a metric of reproductive isolation. And here's what 6 years of data showed us. That is higher than random mating. There's preference for a local male. Although fairly low, it's also uniform. So it didn't vary with genetic distance or phenotypic distance, which defied our expectations. And so not thinking that we were solving the question of this linear and nonlinear dynamic, we decided to extend our studies to include heterospecifics, so further along this speciation continuum. And so recalling that sort of a very simplistic view would be, does it proceed linearly or not? There have been phylogenetic treatments of this group, but there was no consensus to the relationship of these 3 other taxa shown here that co-occur sympatrically, syntopically, and allopatrically to our focal population of red-eyed tree frogs. And so we used the UC phylogeny to understand better the relationship and ask whether reproductive isolation would proceed linearly with respect to these more distantly related taxa. The graph on the right with the speciation continuum, I think this is one of the most beautiful graphs. It's a rank FST of all these pairs of populations, and what it's showing you is that our system has this nearly continuous range of FSTs so that we can really try to fill in those gaps and figure out where tipping points may or may not occur. And so we started with these 3 spots on this continuum of FSTs with these 3 taxa. And so this is what we found. We found really no difference with the conspecifics. They were sort of uniformly high, as we had shown before. But then what's interesting is these heterospecifics. They responded to all of the heterospecifics, although equally, uniformly, and which tells us that this is an incomplete measure, like pre-mating isolation is incomplete at this level, which is interesting, and tells us that we have some more work to do to try to understand those dynamics. We have these gaps to fill. We're going to try to leverage that full continuum to really pinpoint how this metric of isolation proceeds. You know, is there a gradual increase, or do they really follow these tipping points? And so we looked at female mating behavior to measure reproductive isolation. But one of the things that kept nagging at me was this idea that females are trying to convince males to mate with them. Like, they are working really, really hard to court a male, which is opposite of what's supposed to happen. The males are supposed to court the females. And so then I thought, oh, wow, our measure of pre-mating reproductive isolation is probably incomplete. It's probably a muted down, soft version of what's really happening in those swamps. And to really think about pre-mating isolation, I need to include male preference data and male-male interactions. And so this summer, happening right now, we're trying that, and I have a few videos to show you. So these now are live males and females. And the larger females to the right, the smaller males on the left. And they're doing their thing. Now the male's trying to get into amplexus with the female. And then you can see the female move away from the male. And so they're not totally unexpected to see a female reject a male because we just established that there's female preference. But now we have a male rejecting a female. So the female's in the back of the cage climbing up the wall, and then the male is sort of scooting away. And the closer she gets, not interested. And so this is an idea of male rejecting a female. I thought I would give you some satisfaction and show you actual amplexus, so here we go. It was a success for these two. And then the last video is about male-male interactions. And so on the ground is a female, and the male just jumped on, and now a second male is in the cage, and I would also like to get involved. And so now he jumps on. And so now you have this female who made a choice, but then another male came in and decided maybe he would be the choice. So now there's the three of them climbing up the side of the wall, looking for a place to lay eggs. And we're seeing this a lot this summer. We're seeing a lot of male combat interrupting that female choice. And in this case, now the two males are off the female's back, and then the female ends up, if you want to know what happens, that female ends up with the original male. But sometimes she ends up with the other male, so not the one that she originally courted. And so we have this instance of males rejecting females and vice versa, suggesting that maybe mutual mate choice is really important in this system. And that more realistic to the swamps is that we have multiple males in combat, fighting, and that those are going to have a big impacts on how we accurately and holistically measure premating RI. And so it's sort of a call to frog biologists or to other biologists who are working on systems is that this is pretty unexpected. It was unexpected to find evidence of female mate choice or female courtship of males. And so the question is that unusual? Is this a really rare instance of mutual mate choice in a phyllomeducine frog? Or is this just understudied? Have we just not looked hard enough or really thought deeply about the systems that we're studying? And so we're now going to include male and female choice and male-male interactions in our estimates of premating isolation. And we're really excited to do this across the full range of FSTs and capture many independent taxon pairs, fill in those gaps, think about linear and nonlinear dynamics, and also, of course, include more metrics of premating and postmating isolation. The authors on this paper are shown here. This would not be possible. And thank you to our funders and logistic support. Thank you.
We have time for questions.
Any questions? Hi.
Do the frogs see the colors at night?
They see them as shades of gray. And so we did do a visual pigment study and showed that they can discriminate between reds and blues at night in very low light conditions. But they don't see, we don't know if they see in color, but they interpret them as different shades of gray. Yeah.
So a follow-up question, if they see them as kind of shades of gray, do they have another function during the daytime?
Oh, yeah, that's an excellent question. We haven't figured out what it is. If, they might be considered aposematically because they are toxic, but they don't use those colors during the day. They hide them, and in fact, if you disturb them, they like hunker down and hide them more than flash or show them. And so, we don't. That's another unanswered question. Yeah. Thank you.
Hello, everybody. Thanks for coming and sticking to this amazing session. I'm just going to be on this side because my slides are not showing here. So do you hear me well? Yeah, okay, cool. So I'm Sophia Lambert. I'm going to present an ongoing project that I'm doing as a postdoc with Stian Osluka at the University of Oregon, and on unraveling the modes of prokaryotic speciation using metagenomes from a natural environment. So usually in macroevolution, we study species evolution by understanding lineages, going through speciation event and extinction event. And so usually we illustrate that with a phylogenetic tree here. And we assume usually that the speciation process is instantaneous and also that the speciation process is irreversible. But those assumptions are not necessarily always right. And that's why in the field already you've been hearing about the protracted speciation. So the protracted speciation model have been developed to understand the speciation process and essentially to account for the fact that the speciation process is not instantaneous. It takes time to speciate, and it's essentially due to the fact that there are still homologous recombination happening between sister species during the divergence process and diverging into two different species, sister species. And so there is a need essentially to account for microevolutionary processes to better understand the completion of speciation, the timing of speciation, and essentially speciation rates also in microevolution. And so here I'm just going to briefly talk to you about the particularity of the speciation process in prokaryotes. So we've been hearing about spiders and also other organisms, but here I'm just going to focus on prokaryotes. So usually when we think about homologous recombination, we think about sex, but for prokaryotic organisms, well, they sometimes actually behave like clonal reproducing organisms, but sometimes they behave more like sexually reproducing organisms. And this is mainly due to the fact that they have a really different recombination rate. Some organisms actually have a really low recombination rate, and some others have a really high recombination rate. And so this will actually just impact what we said about the completion of speciation. And so in prokaryotic speciation field, there have been several models developed to account for the fact that indeed recombination rate can be drastically different. And so there are two main models that have been developed. The first one is the ecotype model. So under the ecotype model, we assume that the force of speciation, the force of selection is stronger than the force of recombination. And essentially under that model, we will assume that the organisms is behaving more like a clonal reproducing organisms when facing speciation. And so in that case, actually, we can think about the speciation process as being quasi-instantaneous because we don't have this recombination that is kind of delaying the speciation completion process. Under the other type of model, the recombination model, here we assume that recombination force is stronger than the selection force, and thus we will have recombination before actually the selection affects the selection force happens. And so here we are actually having organisms that behave more like sexual reproducing organisms. And what we have indeed is a process that is closer to the protracted speciation process because recombination delayed the completion of speciation. And so the aim of this study here is to actually understand if indeed for prokaryotic organisms we're more in a setting of instantaneous or quasi-instantaneous speciation or more in a setting of protracted speciation. And so basically what we're going to do is look at patterns on the genomes to identify if our data is agreeing more with the ecotype model or agreeing more with the recombination model. And so to do so, we use Prococcus marinus species complex. So Prococcus marinus is part of the most abundant photosynthetic genus in the ocean. So it's really convenient because then we have a lot of genome, and that's what we need to be able to robustly do our inference or estimation. And we know a lot about actually the divergence and the speciation of P. marinus. We know that those organisms are actually diverging through selection, and essentially they are actually diverging through a process that is called niche partitioning. We have actually all the ecotype of the species that are inhabiting a specific depth in the water column, and this is due to the fact that they are actually really adapted to certain light intensity in the water column. So this is really neat for us. And we're going to use the metagenome. So I've told you about the fact that we want to use basically in situ and naturally, like genomes found in the natural environment. So we're going to use metagenomes to look at the patterns that we need to identify which speciation models we're more. under. And so to do so, we use what we call MAGs or SAGs. So MAGs are metagenome assembled genomes, and SAGs are single amplified genomes. So basically, those two types of genomes are reconstructed from environmental samples. But in the case of MAGs, we are actually shotgun sequencing the whole sample and then reconstructing the genome after that. But in the case of SAGs, we are actually isolating cells per cells and then sequencing the genome. And actually, we are also restricting our samples based on their sample site. So we want organisms that are only in oceanic sample site. And so we are actually also discarding all the organisms that have been sampled in the ocean, but then have been cultured in the lab. So some genomes from isolates. And so now I'm going to talk to you about how to identify the genomic patterns that are actually rising from these two different speciation models that I've referred to before in prokaryotes. So essentially what we want to do is detect certain diversity pattern. And I'm going to talk to you about the ecotype model, what's the pattern that it gives on the genomes, and then the recombination model. And to do so, I'm going to use this little cartoon where we have in the circle some individuals from the same species, and on their right we have their genome. And so basically, the little red triangle is a mutation. This mutation is adaptive. It enables these organisms to actually colonize a new niche and have a higher fitness in the new niche. And so in the case of the ecotype model, there is not a lot of recombination. And so the selection will select these organisms. And in case of speciation, we will have what we call a genome-wide sweep. So that means that we have, because the selection have act on these organisms, and this organism can colonize clonally a new niche, then we will have a low level of diversity overall in the genome. And so that's the pattern that we get from this assumption, this model assumption. And so again, this assumes that I can agree with the fact that speciation can be quasi-instantaneous in that case. Under the recombination model, we have a force of recombination that is stronger, and thus we start with the same starting point, but in that case we have recombination. So this gene that is adaptive and has this mutation can actually sweep in a subpopulation, in a part of the population of that species, before getting fixed in a new niche, and thus in the species. So here, in that case, we don't have this low level of diversity overall. We have a regional low level of diversity around the gene that has been selected. And this is gene-specific sweep pattern. And so in that case, again, speciation takes time. So we're agreeing with a protected speciation model. And so actually, just to mention that this pattern can be also due to background selection. And so to differentiate between those two processes, we use a metric that is called the synonymous variant density. And basically, when this metric is low, we have a signal of positive selection. And so we can differentiate between those two. And so actually, just for the rest of the talk, I'm going to focus on trying to identify regional low diversity. So I'm going to try to identify if indeed we have some signal for agreeing with the recombination model, and thus the protected speciation model. And so those are just the expectation to simplify the results we're going to interpret. And so under this assumption that there is no genetic sweep, gene-specific sweep in the genome, we're going to have no difference around synonymous and non-synonymous substitution in terms of synonymous variant density. So here we don't have a regional low level of diversity or regional low synonymous variant density. In the case of a signal for gene-specific sweep, which is again expected under the recombination model, we will have a drop of this synonymous variant density around non-synonymous substitution. And this will be differently affected synonymous and non-synonymous substitution. And as you can see, the horizontal line is x equals 0. So this means like this is a local regional pattern, right, around this substitution. And so here I'm going to show you the results. Are we indeed recapitulating some signal for genetic sweeps in P. marinus species complex? And so again, looking at regional pattern, I'm going to show the result in just one species of this species complex. But basically what we get at the end, again, on the y-axis is the synonymous variant density, and on the x-axis is the distance to substitution. In the middle is 0, and we get a difference, a drop of this synonymous variant density around non-synonymous substitution. And so this difference actually, if you recall the expectation, is a signal for gene-specific sweep. So what does that mean? Again, coming back to the beginning of the story, coming back to what does that mean to, in terms of the speciation process in P. marinus species complex. Well, we have gene-specific sweep in that case, and again, that means that the protracted speciation process might be more adapted to understand the speciation process in P. marinus species complex, and thus that speciation might take also a lot of time in the prokaryotic realm, or at least in P. marinus species complex. And so the idea here would be to also apply this framework to a lot of different diversity across prokaryotic diversity, and to maybe also account for other factors like dispersal rate or environmental fluctuation in some habitat of those organisms. And now we have SSE protracted speciation models, so we can do a lot of fun stuff. And on this, I will thank you for your attention, thank my lab, the Luca labs, especially Signalos Luca, and also Isaac Overcast and Bob Wick for their comments on my slides. Thank you.
Is it possible that ecotype divergence could take time as well in the clonal case?
Ecotype divergence could take time in the clonal case. That's a good question. Well, so in terms of maybe other barriers to gene flows would affect their divergence, such as horizontal gene transfer or stuff like that, maybe. But I actually don't know about the theory around that, but maybe. It's a good question.
I guess I was thinking any multiple substitutions would be required, even if it's
Oh, yeah.
happening clonally, then it still might take a bit of time for the ecotypes to diverge.
Okay. Yeah. Yeah, but it's still comparatively, it will, yeah, probably. But it still comparatively would be way faster, I would assume. But yeah, thank you. Thank you.
And our last speaker of the session, comparing the dynamics of reproductive isolation among clades.
Hello to all. So it's a pleasure to be here, and I really thank the organizer for giving me the opportunity to present the approach we propose for comparing the dynamics of reproductive isolation among clades and its application to an overall comparison between plants and animals. And this is part of the thesis of François Monet, who was co-supervised by Camille Roux and myself at the University of Lille, and by Yves van de Per at the University of Ghent. An overarching goal in diversification and speciation studies is to understand what is shaping the macro versus micro evolutionary dynamics, and we have seen these topics discussed today, and in particular to determine which factors or which traits may explain the differences that we can see among different clades in terms of their dynamics, and also to test for a congruence of the important factors at the macro and microevolutionary scales. So today I will focus on microevolutionary dynamics and present a general framework that we propose for quantifying these macroevolutionary dynamics that will allow to test for difference in dynamics among different clades, and also to identify important factors that can then be compared with the important factors at the macroevolutionary scale. And so the idea is to gather data from many pairs of populations or taxa, and with variable time since they're split, and determine at which phase, for each pair, to determine at which phase of the speciation process they are currently lying. And to simplify, we will distinguish three categories of pairs of taxa. Those that we will call populations, if they are still connected by gene flow, and the gene flow is more or less homogeneous at the genomic level. Then we will define semi-isolated species as taxa which are still connected by gene flow, but with heterogeneous rates of introgression at the genomic level because of the existence of a number of localized reproductive barriers. And then if we don't have any signatures of recent introgression, we will call them isolated species. And so for this, we need a general tool to discriminate among the those three phases, and also we need a universal measure of time because our objective is really to quantify these dynamics. And so to compare to identify these phases of speciation, we propose to use population genomics data, which can be, for instance, two species frequency spectra. And because this allows to detect extent effective introgression by using some proper statistical treatment. And we propose to use ABC analysis because they allow explicit model testing. And for instance, in the data set, we see here a high proportion of shared polymorphism between the two taxa. And so in this case, migration model will be preferred by the analysis. And it could correspond to scenario with a constant migration since the split, CM, or a secondary contact, SC. And in contrast, if in the data set there are lots of fixed polymorphism, then of course it's the isolation models which will be preferred, which can correspond to scenario like a strict isolation since the split, SI, or ancient migration model, AM. And so in this case, we will classify those taxa as species. And in the other case, there is still some ambiguity, so we need to do additional model testing with explicitly contrasting migration model with homogeneous versus heterogeneous migration. And so this type of analysis can be done using the ABC framework, for instance, using the DILS package that has been developed by Christelle Fries and Camille Roux at the University of Lille. So that's for one. And the second thing, that we need a universal measure of time. And for this, with population genomics data, we propose to use the net nucleotide divergence between the two members of a pair, because this is a general measure that can be applied to many different taxa. And so this framework has already been used by Camille Roux in 2016, and he applied it on 61 pairs of animal populations or species or taxa belonging to very different clades within the animal kingdom. And then we can see here the results. Each dot corresponds to one pair of populations or taxa, and the position along the x-axis correspond to the net nucleotide divergence, and the position along the y-axis correspond to the combined probability of the migration models. And so with this, we can, and we use different colors to separate the three groups that I proposed earlier. And so the idea is that this framework can be compared between taxa or between clades, and we wanted to apply it to a plant data set with just to see whether we have similar patterns and also whether the dynamics is comparable. And for instance, we could guess that reproductive barriers may, I'm a little bit in advance, sorry. So one striking result of the Camille Roux's result is that the area of co-occurrence between the semi-isolated species and the isolated species is rather narrow in terms of net nucleotide divergence. And we can see that values are only between 0.5 to 2%. And that was striking given the wide diversity of taxa that were investigated. And so now we have a framework that we can apply to other taxa. And so we want to apply it to the plant data set, and with the idea maybe that reproductive barriers may occur later in plants because of the abundant literature and focus on hybridization in plants. And so our student gather a very large data set of 280 pairs of populations or species from public database, which with the polymorphism data obtained either through whole genome shotgun resequencing or RNA-seq or rat-seq. And we compare, of course, with the data set on the 61 pairs of animal taxa. And so the results are very striking and very simple. The first thing is that for plants, we observe the same pattern that the transition is occur between semi-isolated species and species occurs in a relatively narrow window of net nucleotide divergence, but the overall distribution is strongly shifted to the left. And so it seems that plants stop gene flow more rapidly than animals according to this framework and this data set, and the results are highly significant. So if we look a little bit in more detail the result, so we have here the net nucleotide divergence distribution along the x-axis for the three categories. So if we look at the population on top, there is no difference between the plants in green and animals in yellow distribution. But if we look at the semi-isolated species or the isolated species, we can see that some pairs of taxa start to evolve reproductive barriers much earlier in plants than in animals. And also for semi-isolated species, the distribution of net nucleotide divergence is really shifted to the left. So it really seems that reproductive barriers occur at lower levels of divergence in plants. And of course, it could be because our data sets are not representative. We have many pairs of taxa, but with corresponding to only 24 genera of plants. And also, of course, we are using here population genomics data, so which means that we are focusing on realized gene flow, and we are not obtaining experimentally demonstrated reproductive barriers, which would be interesting to compare, but gathering such data, such experimental data on so many taxa would be quite difficult. So, of course, we then are tempted to speculate a little bit why plants would speciate faster. And it could be because of general genomic differences. And for instance, the fact that plants do possess chloroplast within the cell. And so that this will increase the possibilities of nucleocytoplasmic conflicts. And we know that nucleocytoplasmic conflict may contribute significantly to evolution of reproductive barriers. And also, there is recurrent ancient polyploidy in plants, and we have learned 2 days ago that on average, every angiosperm lineage has witnesses 3.5 whole genome duplication along its history. So this may contribute to an increase in the complexity of the gene networks, which maybe also will increase the possibility of developing Dobzhansky-Miller incompatibilities. And on the other hand, there are also obvious genomic life history differences between plants and animals. Like on average, we can say that plants have more restricted dispersal, which can allow situation of allopatry to arise more easily. And also, maybe there is increased possibility of stronger gametophytic selection in association with pollen tube growth. And also in terms of mating system, on average, plants do possess a larger proportion of species with a mixed mating system or selfing. And of course, within the group, each one has his own favorite hypothesis, but the PhD student didn't want to take position. But the nice thing is that we can also use this framework to test for the impact of some life history traits on these microevolutionary dynamics. And for instance, we look at the effect of life form, and we found that on average, herbs will develop reproductive barriers at lower net nucleotide divergence than trees. And if we look at the effect of the mating system, so we could contrast species with a mixed mating system with species with outcrossing or strictly outcrossing. And overall, we found that reproductive barriers evolved earlier in species with mixed mating system, probably in part due to the more restricted dispersal observed in selfing species. So in conclusion, so we propose a general framework to quantify microevolutionary dynamics that we can be able to test for differences among lineages. Here we choose to compare two very different lineages, but I think it would be probably more interesting to apply it on more closely related lineages, where it will be more easy to understand which factors are important. But however, our framework also allows to test explicitly for the impact of some life history traits, depending whether they are sufficiently variable within the data set. And so this can help also to understand some of the differences we observed among clades. For instance, the effect of selfing. We see that selfing seems to accelerate speciation in our data set. And also it will allow to identify which factors are important and then test for a similar effect of these factors at a higher level of macroevolution. And so with this, I thank you for your attention. And I thank colleagues and the bioinformatics staff for help with the analysis. Thank you. Jonathan?
I was just wondering, in terms of the slope of the models, is that you have used more or less always the same slope? Is there different slopes between the different groups, even that the time for speciation may be different? And also, related to that, the number of substitution gains can be different between clades, and this may be related to different types of speciation.
So regarding the difference between the groups, we can do by genus for each genus analyze. We can test for differences, but it will. So there are some difference, but of course some are trees, some are herbs, and so this is why we need to group them to some extent in order to be able to make explicit tests. But indeed, they will not. It's not like a universal. Everyone is fitting the same thing. So, with respect to differences in nucleotides, so I think here, yes, we are looking at overall patterns. So maybe this is really something that we could investigate whether and maybe to try if we have good estimates. The problem is that most of these data sets we don't have very good estimates of mutation rate or substitution rate, but it could be something that could improve to remove the noise in the analysis. Dan?
Beautiful work. I was wondering if you compared the species-rich clades with clades that are poor, relatively depopulated species.
Which are?
With species-poor clades, did you compare species-rich versus species-poor clades? Poor, with few species.
Oh, okay. No, so for the moment we don't have the data set is big, but still not as big as to be able to do such. So we are not yet at the macroevolutionary scale, but this could be a very interesting to do. I'm afraid. Yeah. Yes.